Hello everyone, this is a lecture on active transport mechanism. What do you mean by active transport? Definition. Active transport is nothing but the movement of substances or molecules against the gradients. It can be against the concentration gradient, it can be against the pressure gradient or potential gradient, electric potential gradient. So the substances are moving against the concentration gradient across the membrane. Consider this is a cell membrane and consider this is a cell. If at all a substance is moving, consider the sodium concentration usually should be very high outside, and because of stimulus action potential generated, sodium would have entered into the cell, and these sodium ions, how they are pumped back to the extracellular fluid is by active transport mechanism against the concentration gradient. So sodium concentration is less inside the cell when compared to exterior. So against the concentration gradient, how the ions are pumped is an example of active transport mechanism. Here you need to understand there are certain transmembrane proteins. The transmembrane proteins are acting like pumps. Transmembrane proteins are those proteins which are through and through the cell membrane. This is the cell membrane and this is the integral membrane protein or the transmembrane protein which is acting like pump. That means they have ATPase activity, and they undergo some conformation changes pumping the ions to and fro against the concentration gradient with a breakdown of ATP. So that is why it is called active transport. Active means there is active role of certain substances or proteins with the help of energy or utilization of energy pumping the substances against the concentration gradient. It is uphill from lower concentration to higher concentration. Transport. So there are two types of active transport mechanisms. Two types. So what are the two types? Primary active transport and secondary active transport. So now we'll start discussing what is primary active transport mechanism. What is primary active transport mechanism with the example of sodium potassium ATPase pump. Sodium potassium ATPase pump. So this is a integral protein or a transmembrane protein which is acting like a pump. So consider this is the cell membrane and the cell membrane is having a integral protein. This integral protein is the pump which is pumping sodium and potassium ions simultaneously. So consider this is extracellular fluid and intracellular fluid. The sodium has to be pumped in or out against the concentration gradient. The sodium has to be pumped out. So sodium has to be pumped out and the potassium has to be pumped to inside against the concentration gradient. So now how this is happening? This transmembrane protein or integral protein will have two subunits. One is a larger subunit and one is the smaller subunit. So larger subunit is the alpha subunit and smaller subunit is the beta subunit. In this alpha subunit, there are binding sites for sodium ions. 1, 2, 3. 3 sodium ions can bind and 3 sodium ions can be pumped. So, and also there are binding sites for 2 potassium ions. And also there is a binding site for ATP. So what happens now with the binding of ATP to this protein and the binding of 3 sodium ions 3 sodium ions will cause breakdown of ATP into ADP and inorganic phosphate, ADP and VI. This inorganic phosphate will bind to the protein and this causes phosphorylation of this protein. So when there is a phosphorylation of this protein, what is happening? There undergoes a conformation change in protein so that the external domain gets opened, releasing the sodium ions. The sodium ions which are bound will be released. So 3 sodium ions are pumped simultaneously from inside to exterior because of phosphorylation, that is breakdown of ATP. Now what happens in the next stage? So the 3 binding sites become emptied and this portion is closed, and there are 2 binding sites for potassium, they are open now. Now what happens? Now these potassium ions can happily come and bind to these areas. They will come and bind to these areas from ECF. Now this PI which was attached, phosphate which was attached to this alpha subunit of the integral protein will be removed. We call it as dephosphorylation. Dephosphorylation by attaching a new ATP. One more ATP is attached, resulting in removal of this PI, and there undergoes a conformation change. What is happening? The outside portion is getting closed, and inside portion gets opened, and inside portion is getting opened. So what happens? Those potassium ions which are bound here can enter into the cell. So this is how 3 sodium ions are pumped out and 2 potassium ions are pumped into the cell by the action of sodium potassium ATPase activity. So here you can understand this pump is a transmembrane protein with 2 subunits, alpha and beta subunit. Alpha subunit is having the binding sites for 3 sodium ions, binding site for 2 potassium ions, and also binding site for ATP, and ATP is broken down by ATPase enzyme activity of the alpha subunit itself. So it is having inherent enzymatic activity breaking down the ATP, and phosphorylation of the integral membrane protein will result in pumping out of sodium ions to the exterior, and addition of new ATP and dephosphorylation of the sodium potassium pump will result in pumping of the potassium ions. So this is how the sodium potassium ATPase pump acts as a primary active transport, pumping out what? 3 sodium to the exterior and 2 potassium ions to the interior. See, now this will not maintain the electrical neutrality. This is pumping more positive to the exterior when compared to pumping less positive inside. What is happening? It is taking out positive. One excess positive is taken out. One excess positive will become outside. One negative will be created inside because of pumping more positive outside. Outside becomes positive, inside becomes negative. So this will create a potential difference across the cell membrane. So this can be around minus 4 millivolt potential difference is created across the cell membrane by the activity of sodium potassium ATPase pump. So that is why this pump is also called electrogenic pump. How much potential difference that is created by this sodium potassium ATPase pump? It is around minus 4 millivolt across the cell membrane with negative inside and positive outside. Minus 4 millivolt potential difference that is created because of pumping of sodium and potassium ions in the ratio of 3:2. If it was 3:3 normal, there wouldn't have any creation of potential difference. That is why sodium potassium ATPase pump is also called electrogenic pump. And what are the sides of sodium potassium ATPase pump? This pump is the transmembrane protein present over the cell membrane. Which cell will have this sodium potassium ATPase pump? The cell membrane of all the cells of the body will have the sodium potassium ATPase pump. So this pump is present in almost all the cells of the body. So that means it should have some important role. Physiologically, it should have some important role. So what is the role of this sodium potassium ATPase pump that is present in almost all the cells of the body? So because of its pumping action of sodium out, more sodium out, so it decreases the sodium concentration within the cell, or it decreases the solute concentration within the cell, so that the cell volume can be maintained. The cell volume is maintained. The cell volume is maintained. Now you consider like when you block this sodium potassium ATPase pump by a blocker, what happens? If this is a cell, this is a cell, the cell inside will have, consider this is the pump or the cell, and this was supposed to pump sodium out more and pump less potassium inside, taking out the solute should be taken out by this pump. Now if this pump is not acting, what happens? The sodium which was entering during the action potential, that keeps on accumulating within the cell. The sodium concentration within the cell increases. So what happens if the sodium concentration within the cell increases? The cell inside becomes more osmotically active, which will attract more and more water into the cell, and the cell swells and the cell lyses. The cell ruptures if at all the sodium potassium ATPase pump is not acting. So that is the reason sodium potassium ATPase pump is present in almost all the cells of the body and pumping out the solutes to the exterior, maintaining the cell volume. So this is about the primary active transport, and why it is called primary active transport. The ATP breakdown is happening at the site of action of pump. See, ATPs are broken down at the site where the ions are pumping out. This is the primary site. ATP is broken down, so that is primary active transport. You will understand when we discuss what is secondary active transport. We'll give one more example for primary active transport. Let's explain with one more example of primary active transport. We'll give an example like consider this is the skeletal muscle, and within the skeletal muscle are the sarcotubular systems, the sarcoplasmic reticulum, and here within this sarcoplasmic reticulum there will be increased calcium levels. And what is the role of calcium ions? The calcium ions which was entered into the cytosol will bind to the actin myosin filaments and cause contraction of the skeletal muscle. Now at the end of the contraction of the skeletal muscle, these calcium ions concentration within the cytosol has to be reduced. How this will be happening? Because of the presence of calcium ATPase pump. So the calcium ATPase pump will pump back the calcium into the sarcoplasmic reticulum. So this is calcium ATPase pump. The action of calcium ATPase pump is to decrease the cytosolic calcium levels, pumping back to the sarcoplasmic reticulum. So this is also an example of the primary active transport, actively pumping out calcium ions. One more example for primary active transport is the parietal cells within the stomach. How these parietal cells will secrete the HCl into the lumen of the stomach. So this is the stomach, consider stomach, and this is the parietal cells. These parietal cells, how these will secrete HCl into the lumen. HCl secretion is by the process of the pump which is present at the luminal cell membrane surface, and this is pumping actively what? Hydrogen ions in exchange for potassium ions. So this is how H plus ions are secreted into the lumen of the stomach. So HCl secretion, H plus secretion is by H plus K plus ATPase pump activity. So here you can understand this integral membrane protein pump. This integral membrane protein pump is transporting one ion in one direction when compared to the ion in opposite direction. So this is like a protein acting simultaneously, pumping two substances in opposite direction. You can consider it as an antipode. So H plus K plus ATPase pump present in the parietal cells. So this is also an example for primary active transport. Now we'll start discussing about what is secondary active transport with examples. Secondary active transport. So we'll give an example and make you understand what is secondary active transport. Consider this is the GI system, stomach and the intestines. Stomach and the intestine. Now we'll expand this intestine portion and we'll expand a cell, intestinal epithelial cell here, and the digested products like glucose or amino acids which are present in the lumen. This is the lumen. How they enter the intestinal epithelial cell. From the intestinal epithelial cell, this side there is a capillary or blood. How it reaches the blood. So from the lumen, how it enters the intestinal epithelial cell, and from the cell how it reaches the blood. Let us explain now. So digested products like glucose or amino acids from the lumen of the intestine, how it enters the intestinal epithelial cell, and from the intestinal epithelial cell how it reaches the capillaries or blood. So the transport of glucose or amino acids from the lumen to the cell is by secondary active transport mechanism. So now we'll expand this and explain. So this is the epithelial cell. This is the epithelial cell. This side is lumen and this side is blood. So now this cell membrane will have a protein, integral protein, which is having two binding sites. One binding site for glucose, the other binding site for one more ion, sodium. So this is a symport. Symport means what? Symport is a transmembrane protein. This transmembrane protein is the symport, which is having two binding sites, so that two substances can bind to the transmembrane protein and simultaneously taking in both glucose as well as sodium. So this we call it as sodium glucose co-transport. Sodium glucose co-transport mechanism. So now here how this carrier protein acts. So before the carrier protein action, you need to understand there should be a concentration gradient for sodium that has to be created between the lumen and the cell. The concentration gradient for the sodium has to be created between the lumen and the cell. So for that there is actively pumping of sodium ions out of the cell, decreasing the concentration of sodium ions within the cell when compared to that of lumen concentration. So now the sodium can enter the cell along the concentration gradient. So from the lumen to the cell, the sodium can enter along the concentration gradient, but this is not a lipid soluble substance. It cannot pass through the lipid bilayer. It cannot pass through the lipid bilayer, but it has to bind to this carrier protein and it has to enter. And this carrier protein is acting in such a way, if at all only the sodium is binding to this carrier protein, this carrier protein doesn't function. This carrier protein will not function if only sodium is bound. So this carrier protein, when it is bound to both sodium and as well as glucose, this functions so that the sodium, this carrier protein undergoes a conformation changes, so that what is happening with the conformation change, the carrier protein outside portion will turn to inside, bringing the glucose and sodium ions to the interior of the cell. So binding of sodium as well as glucose will result in conformation change in this carrier protein, and that will bring the glucose as well as sodium into the cell from the lumen. So now here the sodium is entering whether along the concentration or against the concentration gradient. The sodium is entering along the concentration gradient. Why? Because here the sodium potassium ATPase pump is actively pumping out sodium, and when the sodium is pumped out actively, the sodium concentration in the cell is decreased, and along the concentration gradient sodium is entering. So this is simple, like it's like a diffusion of sodium, but active transport is happening here. So active transport of what is happening? Glucose is getting actively transported from the region of lower concentration to the region of higher concentration. So this is secondary active transport of sodium and glucose, or secondary active transport of sodium or secondary active transport of glucose. Option A, secondary active transport of both. Option B, secondary active transport of sodium. Option C, secondary active transport of glucose. So here this is the secondary active transport of glucose only. Secondary active transport of glucose against its concentration gradient. It is entering the cell from the lumen, and the mechanism of secondary active transport of glucose is sodium glucose co-transport mechanism. So once this